Today we're going to talk about dihybrid crosses. So before we get into that, just a reminder that your genes, the codes for your traits, are carried on chromosomes. The reason we've been using 2 letters for a genotype is because, remember, you have 2 copies of every chromosome. So your cell is going to have 2 copies of the gene for each of your traits. So for example, big P for this particular trait this person has. They have big P on this chromosome, they have big P on their other chromosome. They have little a and little a. They have big B and little b. And so that's why we were using these different letters. Now, so Mendel had a law. He came up with multiple laws, and one of them was the law or the principle of independent assortment. Now, to understand the principle of independent assortment, we have to look at what's called a dihybrid cross in order to understand it. So we've been doing crosses about a single trait. That's called a monohybrid cross, mono for one. What if we wanted to look at 2 traits? So di means 2, and so a dihybrid cross is actually what we're going to specifically be looking at is a cross for between 2 organisms that are heterozygous for 2 different traits. So here's Mendel's law of independent assortment. It says that the alleles of a pair segregate independently of all other allele pairs during gamete formation. Whoa, what does that actually mean? Well, let me show you. So if we do a dihybrid cross, this is looking at 2 traits at the same time, right? So let's say I have 2 plants, and they are heterozygous for red flowers and for being tall. So they have big R, little r for red flowers, but they also are big T, little t. So these are 2 completely different traits. And they're saying that you cross this plant with another plant that's also big R, little r, big T, little t, and you want to know what kind of offspring you're going to get from that. Well, in order to figure that out, you have to do a dihybrid cross. And so that's what I'm going to show you. So we write it like this. Notice how I have the R's together and the T's together. I'm looking at 2 different traits. So that's why I have big R, little r, big T, little t, big R, little r, big T. Like, why do I have 4 letters? Because I'm looking at my R's are for my color, and then my T's are for tall versus short. So I'm looking at 2 different traits. So Mendel's law of independent assortment says that it's completely random. If we want to put it in like simple English, it's completely random which copy of R and which copy of T will be donated in meiosis. Now, why do I have 2 letters here? Again, because one of these 2, when these chromosomes line up in meiosis, and this one carries R, and this one carries little r, and this one carries T, and this carries little t, isn't it true that after meiosis 1 happens, it's only going to get one of these 2 and one of these 2. And it's completely random which one it could be. It could be the chromosome with big R and the chromosome with big T. Here's that possibility. It could be the one with big R and the one with little t. Here's our second possibility, and etc. So this is our third, this is our fourth. So these are all the possible combinations during meiosis when this gives 1 copy of R and 1 copy of T. You cannot put the R's together or the T's together. We're talking about making sperm and egg here. So a Punnett square, if you were going to make a Punnett square of this, you have to show all the possible combinations, remember? So in other words, if I'm big R, little r, big T, little t, and all the possibilities are a big R with a big T, a big R with a little t, a little r with a big T, a little r with a little t. Well, the other parent was also big R, little r, big T, little t. So they could have given big R with big T, big R with little r with big T, little r with little t. And you end up with a 4 by 4 Punnett square with 16 boxes, which is crazy, right? You're like, oh, am I going to have to draw this? There's actually a shortcut I'm going to show you. You would then put your R's and T's back together. Big R, big T, big T, big R, big T, little t. And you would fill in all 16 of these boxes. So I actually have it done already. So here it is. And the results you would get out of your 16 boxes, now if we wrote this in, this is 9331, it's 9 out of 16. So out of all 16 of my boxes, 9 of those offspring would be red and tall. And I could go through and find them. Red and tall, 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 red and tall. And that's it. 1, 2, 3, 4, 5, 6, 7, 8, 9. There's my 9 out of 16 that would be red and tall. 3 out of the 16 would be red and short. 3 out of the 16 would be white and tall. And 1 out of the 16 would be white and short. So this is how you solve it with a Punnett square. You have to come up with all the possible outcomes and then make a Punnett square that's big enough to accommodate what you could get from that cross. This is showing what they mean by independent assortment, that it could be the blue and the blue and the red and the red, or it could be the big blue with the little red or the big red with the little blue. That's the reason we have to write so many possibilities. Now, what if we cross two purebreds? Well, let me show you. So, big R, big R, big Y, big Y. What can this one give? Only big R and big Y. That's it, right? There's no choices. And the other parent, little r, little y, can only give little r and little y. Remember, we have to put 2 letters because we're looking at 2 traits. And if we fill it in, it would only be literally a 1-box Punnett square. All the offspring would be heterozygous. They would all be, oh, sorry, this isn't for red. This is for round. This is for round and yellow. So they would all be round and yellow. So you would only need 1 box. You would not need a gigantic Punnett square. This is why I told you it would be so useful for you if you understood how to make these Punnett squares different sizes. And then this is showing, again, a dihybrid cross where they're both heterozygous. So they're crossing the F1, big R, big Y, big R, big Y, little y, and you had to write all 4 possibilities for this parent, all 4 possibilities for this parent, and fill in all the boxes. All right, so let me show you one more with a Punnett square, and then I'm going to show you a shortcut with something called the product rule. So let's look at this. So big B is for black. I hate this one because they have big L for short hair, which is kind of confusing. You'd think L would be for long, but it's not. So our 2 parents are big B, big L, little l. Well, what can that parent make? Well, they can make big B with big L, big B with little b with big L, little b with little l. So this is what we would have to write here is all the possibilities for this parent. So big B, big L, big B, little b, big L, little b, little l. Now, here's the shortcut, my first shortcut. My second parent is not heterozygous for both traits, right? They're little b, big L, little l. There's not 4 different things they can make. They can only make 2. They can do little b with big L or little b with little l. That's it, right? They don't have a big B. So technically, we don't need this. We only have little b with big L, little b with little l. So this would be our Punnett square right here like this. So you get big B, big L, big L, big B, little big L, little l. Little b, big L, little b, big L, little l. And you'd have to fill in all these boxes. Now this is still tedious, so I'm going to show you a shortcut. That's actually much shorter than this. This is what I do with AP Bio, is this shortcut. Now if you sort this out, our offspring are actually going to be out of 8. So if you literally count it up out of 8, you're going to get 3/8 black and short, 1/8 black and long, 3/8 white and short, 1/8 white and long. All right, so now let me show you this shortcut. So let's say we do this same cross, but I'm going to show you something called the product rule. So the cross we were doing, I'm going to switch over to my other board here. Okay, so the cross we were doing was big B, big L, little l, crossed with little b, big L, little l. The product rule says that the chances of any 2 different things happening is the product or multiplying their individual probabilities. What in the world does that mean? Well, here's what we're going to do. So instead of making the big Punnett square, we're going to separate out our crosses and just cross our B's and then just cross our L's. So if I just cross my B's, I'm going to get a 1/2 chance that they could be black and a 1/2 chance that they would be white. Now let's cross our L's. So we're crossing big L, with big L, little l. And that gives me a 3/4 chance. Remember, in this case, even though L, you would think long was dominant, short is dominant. So this is actually a 3/4 chance of being short and a 1/4 chance of being long. So all we've done here is we've separated out our traits back into just simple Punnett squares. So we're just doing our B's, we're just doing our O's. But if we want to know the chances of black and long, the product rule says multiply, get the product of their individual probabilities, right? So what's the chance of black? 1/2. What's the chance of long? We just calculated 1/4. What's the chance of black and long? 1/8. Now let's get the chances of black and short. So the chances of black are 1/2. The chances of short are 3/4. 1/2 times 3/4, 3/8. Now let's do white and long. It's going to be 1/2 chance of white times a 1/4 chance of long. And that's going to give me 1/8. And then white and short, 1/2 chance of white, 3/4 chance of short, 3/8. If you do this right, these should all add up to 8/8. And that's how you can do these same crosses with the product rule. Look how quick it makes the original cross. So remember the original one we were doing, it was 2 heterozygous ones, right? Big B, big L, little l, big B, little b. This was the one with 16 boxes. Well, if we just separate our B's and our L's, we weren't doing it with B and L, just FYI, but let's just say we did, and they were both heterozygous. And I want to know, hey, what are the chances in this cross of black and short? So instead of doing 16 boxes and counting how many are black and short, if this was my parents, I could just do the black. My chance of black is going to be 3/4, if you fill this out, because I'm going to run out of time on this recording, so I'm just going to check off these boxes. And then my chance of short was actually dominant, so that's 3/4. Guess what? Chances of black times the chances of short gives me 9/16. The same exact thing we got when we did it with the 16-box Punnett square. So this is called the product rule. And again, the way you use it is you still have to figure out what the original genotypes of the parents are. But then instead of trying to figure out those big Punnett squares, we have to put 2 letters over every box. You can separate out the individual crosses, get the fraction of offspring that should show each individual trait, and then get the product or multiply those, and that will give you the fraction that should have the trait in the end. All right? And so that's how you do a dihybrid cross. I hope that the shortcut is helpful for you. See my 9/16, 3/16. Again, with the product rule, we would just separate this out and do our R's, and then do our Y's, and then get the fraction of round versus wrinkled, get the fraction of yellow versus green, multiply the fractions together. All right, so next time we're going to talk about pedigrees, human pedigrees. How do we track traits over time and kind of figure out, hey, did this come from the grandma? Did it come from a great-grandparent? Is a trait dominant or recessive based on whether your parents have it or not? So that's going to be our goal next time.